Hello and welcome to the Mind Report. My name is Jonathan Phillips from Yale University's Departments of Philosophy and Psychology. And today I'm talking with Randy Gallistel from Rutgers. Hey, Randy.
Hi, Jonathan.
Hey, it's great to have you here.
It's great to be on.
Good. So we've got a lot to talk about today, and I'm really excited about it. But I thought maybe it'd be fun first if we could just talk a little bit about your own background, and maybe that will give everybody a sense of sort of where you're coming from.
I was trained as a behavioral neuroscientist at Yale in the physiological, what we called it physiological psychology in those days. That was in the 1960s. And that was after my undergraduate work at Stanford, where I did animal learning research and brain stimulation work under the direction of Tony Deutsch. And from Yale, I went to Penn. I was there 23 years. And then my wife and I went to UCLA. We were there for 11 years. And then in 2000, we came to Rutgers, where until recently we were co-directors of the Rutgers Center for Cognitive Science.
Right. Yeah, this does quite a background. And the whole time have you been doing sort of research on brains and how they function, or has your focus sort of shifted over time?
Well, I guess so. For almost 30 years, I did the psychophysics of rewarding stimulation of the medial forebrain bundle in the rat. And the goal was to try to identify the particular set of axons that was being excited that was causing the rewarding effect. And I thought that would function as a nerve memory preparation, because you're producing an input to the brain by direct electrical stimulation, so you have unparalleled control over the signal that you're generating in the brain. And the animal remembers, as you vary the parameters of that stimulation, you vary the magnitude of the reward, and the animal remembers that magnitude, and you can easily measure that memory in its behavior.
Right.
So I thought that we'd find the axons and we'd follow them to the place where memories are made.
Yeah.
We were never able to conclusively identify the axons. It was very disappointing, but it was very interesting work in many other ways. Recently, one of my students, Peter Shiskal, has begun using this optogenetics technique, which allows you to select which of the axons in a complicated bundle like the medial forebrain bundle, which of the axons you will actually excite with your stimulation.
Right, so you can sort of causally intervene and excite some specific axons.
Exactly.
Yeah.
And so I have hopes that we will finally identify them. But in the meantime...
The answer.
Yes. In the meantime, for one thing, I couldn't get funded in that work after a while, along about the end of the 90s. And so I thought, well, how else could we get at the physical basis of memory? And I was extremely impressed with the strategy that Seymour Benzer used in getting at the molecular and cellular neurobiology of the circadian clock, which when I was a graduate student, the circadian clock was a total mystery. A lot, like all total mysteries, a lot of people didn't believe there really was a clock. They thought it was some sort of emergent phenomenon, right? The usual nonsense when you don't understand.
Right.
And Benzer said, well, look, I think there's a clock, and if there's a clock, it must have some modest number of genes that determine the basic structure of the clock, and so I'm going to start looking for, behaviorally, for mutant Drosophila whose clock isn't functioning properly. Right. And Max Delbruck thought he'd lost his mind, because I think Max didn't think there was a clock, or God knows, or if there was, you could never find it genetically. Anyway, as I think you know, as now a famous story, Seymour succeeded spectacularly, and rather quickly. So they found mutants that had abnormal periods in the clock, either too long or too short. And this was really the keys to the kingdom. Now we know a tremendous amount, and we're learning more every year about the molecular and cellular neurobiology of the circadian clock, and it's in every textbook and so on. So, like a lot of other people, I think, including Benzer, I think that same strategy could be applied to go after other basic mechanisms like memory. And in fact, Benzer himself also tried to get at memory that way, I think much less successfully.
Yeah. Okay, so I mean, a lot of your work, and this is exactly what we're going to get into, is about memory and how memory might function in brains. So, but I think it might be helpful for everybody if we set up the debate a little bit. So the issue that we're really going to be talking about is an issue about sort of how the brain does what it does, right? So on the one hand, we know that the brain is, say, made up of neurons or axons, and we know that somehow this must be part of the brain that allows us to perform these fantastic feats of memory that we have. So, for instance, we're able to remember sort of thousands of different faces, even though many of them may look similar. We may be able to use to sort of wealth of information to sort of locate ourselves in space and to get from one place to another. And the debate that you're particularly engaged in is this one about what sort of computations is the brain doing to allow us to be able to do that, and how is that instantiated in the brain?
Yes, that's very much true. But I indeed am involved in that debate. But to me, the inner kernel, the single most important issue is how we conceptualize the physical basis of memory itself.
Right.
So that's what this book that you mentioned is really about. But to understand what the argument is, you have to know a great deal. And so in that book, we're trying to explain everything that we think people know in order to grasp what the problem really is.
Right. Yeah. So, and you do I have to say, you do a fantastic job of setting all that information up. And I mean, it's a lot of fun to read in some sense, because I think you guys do sort of give the reader all of that information. But it might be so much information that you can't totally cover it all.
Yes, I'm afraid that's a problem.
So I think that the side that we do want to focus on, and we can talk about, is sort of exactly the part that you picked up, and this is the part of what is the brain sort of physically doing when we're encoding the memories that allows us to accomplish the feats we're able to accomplish, or say that other species are able to accomplish. And so I think we'll have to I do think everyone should read your book, but we might have to focus here just on the sort of bigger picture and that debate in particular, rather than information theory or signaling.
Sure, of course.
Good. I was a little worried that we were
Oh no. I don't think a blog is the place to go over information theory.
Yeah.
Important as I think it is, but
right. So maybe a good place to start is just sort of by admitting that on some level there's so much about neuroscience in the news, and so much there's always new discoveries that are being made, and in some way we know a lot about what's going on. There's all sorts of articles with pretty pictures of brains. But on another level, we actually don't know what the brain's doing. So we don't actually know how it's, say, storing memories.
That's God's own truth.
Yeah.
We don't. And we've been this is a problem that many of the leading figures in behavioral neuroscience, certainly, and more generally neuroscience, have worked on now for more than a century.
Right.
So the fact that we still don't know is frustrating.
Yeah, right. So you might think about it like, I think I found this when I first began learning about this sort of analogy to help. So you might think about it sort of like maybe as a computer. So suppose that some alien came down and they found my MacBook, and then they were going to study it, and they pressed these buttons, and then sort of YouTube videos came up, or they could type things on Microsoft Word, right? But and then they also could sort of look at the actual boards or the gates, and then they sort of had an idea of what the structure was, but they really don't understand how it is that this physical thing is causing the things on the screen to happen.
Indeed, yes.
Right. And so you're really arguing I mean, and this is exactly what the debate is. So what is happening when instead of the circuit board, we're worried about brains, and you're really arguing against a particular understanding, which is very accepted in neuroscience about
Yes.
what the brain is doing. So maybe you can actually just give us a sort of an overview of what that view is.
Well, I think particularly, I think you're in philosophy, and the easiest way to understand it is that the view that neuroscientists currently entertain, they inherited fairly directly from the English empiricist philosophers. It's basically the ideas of John Locke and David Hume and Bishop Berkeley, translated into neurophysiological language. So what used to be ideas have now become neurons, and
Yeah.
but of course, it's a cartoon version of what these Locke and Hume were much subtler thinkers than I will be giving them credit for in what I'm about to say. But really, what's been translated into neurobiology is a kind of cartoon of what they were thinking. But so reduced to a cartoon level, Locke's idea about how the mind works was, look, you have these ideas, and there are elementary ideas which are directly caused by stimuli, by we call those sensations.
Mm-hmm.
And if two of these ideas often occur together temporally, then some sort of connection between these ideas grows up, and then this explains the train of thought, if you like, in that one of these ideas that's connected to another idea, it happens to occur for whatever reason. And because of this association, because of the connection between the ideas, the occurrence of idea one summons up idea two, and so on. And so this story, of course, has had enormous appeal now for several centuries. And this story has been translated fairly directly into neurobiological concepts, almost by word substitution. So instead of let's substitute neuron for idea, right? And so when one neuron is activated, and if another neuron is repeatedly activated at the same time, I'm leaving out some of the details. They have to be sort of pre-connected to each other in some way we needn't go into. But if these two neurons are repeatedly active at the same time, then the connection between them becomes stronger. And so now when one neuron is activated, it excites the other neuron.
So just like, if you had the association with the idea of salt, and then when you think of salt, you're likely to think of pepper.
Exactly.
When this neuron gets excited, then this other neuron also happens.
Exactly. In fact, I've been arguing against not only the neurobiological interpretation of this view, but also the psychological. That is, as you well know, in both philosophy and psychology, ever since the 17th century, associative theory has played a very strong role, and I've been arguing now for more than 20 years without much success that the association is the phlogiston of psychology and philosophy, that an association never formed in the brain of any animal. And your salt and pepper caused a smile because when I was giving a version of this talk, 10, 12 years ago perhaps, a very, very eminent psychologist of the associationist persuasion, I won't name who it was arguing with me afterwards, and he said, well, how come if you say salt, I think pepper? So, as far as he was concerned, this was pretty much definitive proof that there must be such a thing as an association, right?
It's not the most sophisticated argument, but I definitely that one example. I mean, you can feel compelled by it in the right moment.
I'm sorry?
That particular example, I think it's easy to feel compelled by.
Yes, exactly. So this is really this, if you like, is the computational theory that underlies the thinking of most neurobiologists about memory. So they think memory is they call it the Hebbian synapse. A Hebbian synapse is a connection between neurons that behaves in the way I just described. That is, if the neuron on one side, on the downstream, so to speak, side of the connection becomes active at about the same time as the neuron on the upstream side, then the connection between them, the synapse between them, becomes stronger. And that's called a Hebbian synapse, and it's an extremely straightforward translation into neurobiological entities of the basic conception that John Locke was arguing for 300 years ago.
Right. So on this view, it's basically that when we're thinking about, say, about learning or how we're storing memories, basically, I mean, when I learn something, all I'm doing is just changing the strength of these connections between neurons.
Exactly.
And that's just physically realized in my brain. So, I mean, that just is what learning is. There's nothing more to it than
you can read some version of that in the New York Times science section at least every month or two.
Right.
Whenever they're talking to a neuroscientist about memory, that's the story that they tell you.
Right. Okay, great. So we have this view, and it has its roots long ago, and now we've translated it into neurobiology, and it's widely accepted. So your book is a lot of it is about what's wrong with that
Yes.
view, and so I want to see if you can. I mean, of course, you won't be able to go into all the details, but I think you have some really compelling examples with different types of animals, and I think you have a more general sort of. I think there's hopefully there'll be a way to sort of summarize what you think is wrong with this approach.
I'll try. I would try by going back to what, in some sense, everybody knows. I mean, your grandmother, if you ask her, well, what's memory? She says, well, when you learn a fact about the world, you store it in memory, right? I mean, that's what memory does. It records the things that you learned.
Right.
Now, this story that we're discussing, the associative story, actually doesn't explain how facts are stored in the brain. And you can see this very clearly. As you know, I like to focus strongly on the navigational performance of animals of all kinds, but particularly insects, who are much more spectacular navigators than most people realize.
More than most people.
Yeah, well, to some extent. Because navigation depends fundamentally on remembering facts about the world, right? You have to remember how far X is from Y and what direction it is from Y. And I've been approaching my fellow neuroscientists in this business. I mean, of course, I've been in this business a long time, so I know most of the major figures who are working on the neurobiology of memory. And I keep asking them, so how do you store a location in the memory, right? I mean, what do we mean by a location? Well, we mean a vector. We mean two numbers, a latitude and a longitude.
Right.
Every scheme for encoding location has that property. No one knows how to encode a location that isn't some version of that vector story. Even the grid cells are, which I won't bother to explain it, but they're a version of that story. So you would think that if the changes in synaptic conductance are what memory is, then you would get a nice, clear, snappy answer to how it was that the location was encoded in changes in synaptic conductance. And I'm here to tell you, you will not only will you not get a nice, snappy answer, you will get nothing but smoke and mirrors. You will get, it's an emergent phenomenon. Oh, it isn't really at the synapse, it's in the whole circuit. And the hands will begin to wave and the smoke will begin to blow. And then you'll say, I don't think these people know what they're talking about.
Good. So let's go to your example of dead reckoning in ants. I think is sort of the best example of this exact thing that you're talking about.
It's a good one.
Yeah, and so maybe you can tell us first what dead reckoning is and sort of how it works, and then particularly about the ants and the studies that have been done on them, and you can sort of see why even in an ant, we really need this sort of location.
Yeah. Well,
Yeah.
so dead reckoning is the if you go to take a course in navigation. Before the days of GPS, but even now they teach everybody in the Navy this because they think someday they'll be in an open boat without their GPS, and they need to know how we used to do it,
Yeah.
right?
Yeah. And the batteries die.
It's how you keep track of at least approximately where you are when there are no familiar landmarks that you can use to judge your location by. And of course that happens as soon as you sail out of sight of land, right? And once the land has disappeared behind you, you look around, it's all just waves. Right. And one way, the waves are not landmarks; they don't stay put. So how did Columbus know where he was? Right, he started out from the Azores, and he headed out into the open ocean. He had a map of Europe, but he was headed into completely unknown territory. Well, he was sailing more or less due west, and the kind of boat he was in, under the trade winds he was sailing in, it made 100 knots, 100 nautical miles or something like that every day. So by keeping track of how fast the boat was going and making an entry in your log, they used to do it every half hour, you would, that's when the bell rang, you would make an entry as to how fast the boat had been going during the last half hour and the direction in which it was going. Right. And now, it's not rocket science. Suppose I leave New York and I'm sailing east and I'm making 5 nautical miles an hour. So when I leave the Ambrose Light behind me and I sail for 1 hour, where am I roughly? Well, I'm roughly 5 nautical miles east of the Ambrose Light, right?
Yeah.
And after 2 hours, I'm roughly 10 nautical miles and so on, right? Now, of course, if I start sailing north, now I'm going to have to do a little trigonometry, right?
Yeah.
But the thing was, you kept this set of entries of your velocity. That's basically what you're recording here. And then, of course, velocity times the time equals your displacement, that is the change in position. And if you add up all the changes in position over each half hour, that's your net change in position. And that's an estimate of where you are.
Yeah.
And there's a very extensive and quite lovely literature, experimental literature on animal navigation, particularly insect navigation, because ants and bees lend themselves exceptionally well to studies of navigation. There's one thing that there's complete agreement about within the field, based on extremely compelling evidence, that the ants and the bees, and for that matter all God's critters, homing pigeons and migratory birds and even human beings, are keeping track of where they are in no small measure by doing this process that I just described, by dead reckoning.
Right.
And what's important about it from brain theory is that you ask, well, what are the essential features of a machine that could do this? Well, it has to have a memory where it keeps the sum of the displacements, right? Because that's your record of how far you've traveled, of where you are, basically.
Right. You have to store that.
You have to store it.
Number.
I mean, if the logbook in which they were writing it all down washed over, bored or burned, well, you were relying. You hope some members of the crew have been reading the logbook and they remembered where we were, right? Because you need a memory, you need a record, and the record has to be accessible to computation. This is fundamental. I can't stress too strongly how important this is.
Yeah.
That is, you have to be able to pull it up and add to it, right? If you sail another half day, you have to say, well, look, here's where we were this morning, and now we've sailed for 6 hours, and we've been sailing east at 6 knots, let's say. So we're 36 nautical miles east of where we were this morning. So you have to be able to take out the number that represents where you were or actually the vector, where you were this morning, and add your most recent displacement to get a new number, which is your new position, your new displacement, so on. So the ants, so ants are doing this. Now, ants have less than 1 million neurons in their brain. There are fewer neurons in the brain of an ant than there are in one voxel of an fMRI recording from a human being.
Right.
And many imagers seem to think that in one voxel practically nothing is happening. Well, in the brain of that ant, and he has a lot of things to do besides keep track of where he is. But keeping track of where he or she is fundamentally important. If the animal doesn't know where it is, it's as good as dead. And they're doing this dead reckoning. And there's just no defensible story out there about how they're storing their current position and how they're adding to the current position.
Right. So just to give everybody a sense of and I think it's a really fascinating phenomenon of what the ants are doing and what makes this kind of impressive. The particular ants that are mostly live in just a sort of a desert.
The Tunisian desert, yeah, they've been studied by Rutgers in Switzerland. Fabulous work. Every summer they go there and they follow these ants around in the Tunisian desert. It's it's 130 degrees.
Right. Right. So, and then in the desert, in particular, I mean, it doesn't have the sorts of landmarks you might be able to rely on.
The desert for an ant is exactly like the ocean for a human being. It's
right.
It's all pebbles.
Yeah, exactly. So they go out and they leave their very small their nests are just sort of a small hole in the ground, not much bigger than the ant itself.
Yeah, right.
And that's the opening, and they go out into the desert and they just wander around, pseudo randomly, looking for food or something.
That's right.
And they may go quite.
They
quite
go a long
distance.
ways, 50 meters, better part of a football field.
Right, in all sorts of different directions, but once they find the food or whatever they're looking for, they take a direct line.
They turn around, they go straight home.
Straight home, and sort of not missing a step, as it were, or not getting.
Well, they know the direction home with astonishing accuracy, particularly given that the route they took out was often unbelievably torturous. I've showed the routes these ants had followed on the way out to sailors, and I said, could you follow a route like that and know where you were with any accuracy? And they just laughed. They think, no way, I'd be lost.
Right.
But the ant knows the direction home, and it knows the distance. Experimental work shows that if you move the ant so that it's running to a hole that isn't there, the ant runs until it should be basically at the hole, and then it stops and starts looking for the hole.
Right. So it's sort of calculated not only the distance but the direction it needs to go in, and then it goes directly that far. And then if it doesn't find the hole, it thinks something's gone terribly wrong.
Right.
But to be able to do that, it has to do exactly what you're saying. So it has to store.
Numbers.
Dead reckoning. Yeah.
I mean, you ask people, so how do we record direction and distance? Well, it always boils down to numbers. One of the things that it's really hard to get people to understand is one of the great lessons of modern technology is that if you have a machine that can store a number, it can basically store anything whatsoever.
Right. Right.
So the question is, how would you store a number in a Hebbian synapse?
Yeah.
Try out that question on your neurobiological friends and see what answer you get.
All right. And I think this is
mirrors.
and I think this is, I mean, it's actually, we're talking about it on a very theoretical level of what sort of computations brain do, but it actually bears directly on a lot of things. No, and I mean, one thing you might just think on a very practical level is, I mean, I don't know if you've heard about Obama's new initiative for the brain activity map.
Yeah, oh, sure, I've heard about it.
Yeah, and so he's just, I mean, I think like a day or two ago, he just said, we're going to give the first $100 million to it. We're projecting something like $3 billion over the next 10 to 15 years. And this is the thing that's supposedly going to solve, this is going to solve I think he said something like, it was something, I mean, it was a little bit grand, but I think he said something like, we've seen things smaller than an atom, and we know the mysteries of the universe, like light years away, but we haven't unlocked it's like 3 pounds of matter or something that sits between our ears. And,
well, he's right about that.
He's right that we haven't unlocked it, but I mean, the approach that this particular project is taking is to,
map the brain,
to look at the connections.
It's even worse in Europe. The European Union is allotted $100 billion, €1 billion to a similar kind of effort.
Right. So I mean, we've been talking about this, but your basic view is that approach is just smoke and mirrors in some sense. It's not going to give us the thing that even an ant has to
I'm
be
afraid
doing.
it's not. No, because we don't know. You in your opening remarks, you mentioned suppose the Martian is looking at the insides of a computer.
Mm-hmm.
A computer technician can look at the insides of a computer and make sense of it because the computer technician has a computational model of this machine. He knows what the pieces are, and he knows what each piece does, and he knows how it does it, and he knows how they relate to each other. And that
Right.
it's that computational level theory that enables one to make sense of the connections. If someone who doesn't have that level of conceptualization of the machine they're dealing with looks at this spaghetti inside, it's it's spaghetti.
Right.
There are a lot of
Right.
connections there. They must be doing something. It's likely that this part is probably doing something different than that part, but we don't know what exactly the parts are. We don't know what they're doing. We don't know. We don't know how they in one sense, we don't know how they transfer information between the different parts. There's a tremendous amount we don't understand.
Right. So it'd be to go back to the computer example, which I think is helpful here. So your view is basically, it would be as if we had a board and we didn't know a computer board and we didn't know what it was doing, but we knew we saw YouTube videos and sometimes it ran Microsoft Word. And now we want to know why is it sometimes doing one or the other. And now we're going to propose something like $100 million to just really look at the board a lot, to really just look at
That's
the
right.
different connections on the board.
Yeah.
But you might think that's never going to tell you.
Well, there's actually kind of a case study that helps you to see the cerebellum is a part of the brain, a rather largish part of the brain, that has a particularly simple as things go in the brain and repetitive cellular structure to it and connectivity. So that when you look at this structure, which we've known a lot about now for a very long time, you think, damn, if I was just smart enough, I could figure out what the cerebellum is doing just by looking at the way in which these neurons, because there's this tremendous amount of pattern here in the way in which they're connected. It's very repetitive. Well, I remember listening to, on more than one occasion, to talks by John Eccles, a very famous Nobelist in neuroscience, neurobiology, a student of Sherrington's, famous electrophysiologist, who in his last decade or two, he lived a very long time, worked on the cerebellum. And he was not a modest man. He was quite a good speaker, but a lot of trumpet blasts at the beginning about things. And so he said, we got this neural machine, and we understand how this neural machine works. And he would describe this lovely anatomy that we've known about now for quite a long while. And then he would describe the recordings that he'd done from the electrical activity and so on. It was all a lovely story. Mm-hmm. But when you walked out, he said, well, wait a second. He never said what this machine does. It's a little bit like someone bringing you an internal combustion engine and not telling you that it's an internal, that it's a power plant, right. And saying, look at this incredibly repetitive anatomy, and look at this little piece that goes up and down, up and down, up and down, and explosions occur here, and so on.
Right.
And you say, oh, that's all very fascinating, but what does this machine do? And can you be said to understand a machine if you don't know what it does and how it does it? Well,
right.
The fact is, we to this day, so the talks by Eccles that I'm talking about go back 25 years now, we still don't know. There's no agreement in neuroscience about what the cerebellum does. Not even
right.
the beginnings of a consensus about what the cerebellum does. There are wildly different theories about what it does.
Yeah. Right. Yeah. And so you might think that if we can't even figure out what the cerebellum in general is.
If we can't figure out the cerebellum from the connectivity, the hypothalamus, the cerebellum looks like a crystal. When you get to the hypothalamus, it looks like spaghetti. It's a god-awful mess.
Right. And so, yeah, and then another way of thinking about, this $100 million that was just given for the mapping sort of the structure of the brain is we'll say, to look at, what are other cases where we have done that, right? So what are cases where we've actually mapped out all the neurons and their connections to each other?
C.
And
elegans.
did that give us an answer?
There's this worm, a little worm that has 304 neurons. It has about 1,000 cells, and 304 of them are neurons. It's one of the smallest creatures with a central nervous system. And it's been studied extremely intensively over the last 30 years, developmentally and from every other perspective. And so we know the developmental history of every single one of those neurons, and we know the synaptic connectivity of every one of those 304 neurons. And the C. elegans has a fair amount of rather complicated behavior, and we've hardly begun to understand how it is those neurons actually generate that behavior.
Right. Yeah. So you think, if we're going to understand the what's between our ears with this approach, with this C. elegans, we've done it, right? So we don't even have to work.
We found out all the connectivity. We still don't know how it works. Don't get me wrong. It's very important to know the connectivity. It's not as if this is useless knowledge, but it isn't going to tell us how the brain works.
Right. Because we can't even predict the movements of C. elegans now that we know that. It's not giving us the answers. Yeah. All right. I think that's good. It'll be really interesting to see. The next 10 years, neuroscience very much seems to be headed that direction, whether you like it or not. So it will be interesting to see, what happens there, and if they do begin doubting or they begin changing their methods. Or, of course, it's possible that. It's possible that maybe they will find the answers. No one has ruled out that possibility. There are ways maybe that one could go.
It's possible. Not likely,
but maybe
possible.
You think it's not very. Yeah. So one thing in the book is that you compare this idea of that we need to be understanding sort of how the brain is storing and updating values of variables, like we talked about in the navigation case. It's somewhat like DNA. Right. So before, now at least a while ago, we didn't really understand exactly what was giving rise to how things grew or what exactly happened. But we had this idea that something must be responsible for it. There must be some structure there that's making sense of it, and it must be doing something like this.
Yes. I'm sorry. Is that the?
Well, yeah. You can pick up. I think you know where I'm headed there.
I think that. There's a wonderful book on the history of the molecular biology of the DNA by Horace Freeland Judson called The Eighth Day of Creation, which I've been recommending very strongly to people for years. It's a fascinating story, brilliantly written, and the discovery of the molecular identity of the gene, the molecular structure of the gene, was arguably the most important scientific discovery in my lifetime, and certainly way up there with the top 10 scientific discoveries and insights of all time in any sciences whatsoever. And I think it's full of instructive lessons for where we are now in neuroscience, with the neuroscience of memory, because before Watson and Crick identified the quite unexpected structure, people really, the gene was a huge mystery. It didn't make biochemical sense. It was so puzzling from a biochemical perspective that many biochemists didn't believe there actually were genes. They thought it was, the usual nonsense emergent phenomenon and so on. It emerged from some sort of complex, messy biochemistry that we would never really understand, and so on. But there were others that said, no, there really are genes, and we should try to think what the biochemistry is. And there were a lot of people who tried to make up theories, and none of them were even close. Right? None of them even had the right idea, right? That you had this digital code by the sequences of nucleotides. Even though in retrospect, there were some thundering clues out there, like the Chagraph ratios that in retrospect, when you look back, it was like going back and starting an Agatha Christie or a mystery book all over again. I think, oh man, I should have picked up on
How did I
that
miss
clue.
that?
That was a huge pointer. But no one did. And of course, since it was basically impossible to spin out a plausible biochemical story, many biochemists said, well, genes are just a fiction of the geneticist's imagination. Geneticists, they're not really serious scientists like us biochemists, and so on. But in fact, the geneticists were busy working out a very detailed computational story about the gene in the total absence of any knowledge of what the physical structure of the gene was, right? But they were able to measure how far apart genes were. They were able to figure out how many different independently assorting sets of genes there were, and then you could say, oh, look at that. Every species has as many independent assorting sets of genes as there are chromosomes. Whoa, that's pretty suggestive, isn't it? Looks like the gene, whatever it is, it must be in the chromosome, right? So there was a very detailed computational story that the geneticists had worked out. And when you read this Eighth Day of Creation book, you see that computational story was driving the molecular biology at every turn. If you took away that computational story, if they just said, well, we're gonna figure out the molecular basis of heredity, but we have no computational story to tell, the whole effort would never have gotten off the ground. It would have just been floundering around, barking up the wrong tree, and so on.
Right. And so you might think that similarly, the thing you're pointing to is that, I mean, not that you have the answer that it must have the structure, maybe like they didn't at some point, but there has to be something like this. It has to be, it has to have these sorts of properties. It has to be able to do this.
Jonathan, you're getting a little hard to hear at my end, are you?
Oh, sorry. Is that any better?
It's still not very loud.
Still not very loud.
That's a little better. There we go.
That's a little better. Okay, let's try this. So the thought is that similarly to DNA and figuring out what are the things that DNA has to do, you're saying, look, we don't know exactly what the process is, but we know it has to do these things. We know it has to have these sorts of features.
Well,
but then
sorry, go on.
Oh no, but then you might think that there's one disanalogy is that you might think that one disanalogy is that in some sense, what allowed us to discover that the exact structure of DNA was that we got better tools, right? So we were able to sort of see further down. We got really good at looking at smaller things. We were able to work out how replication was happening and things like that. I mean, do you think that there's something similar that's going to happen in this case that's going to allow the sort of breakthrough that you're saying we really have to have?
I think... by the way, I just discovered why you were getting harder to hear. My earphones were slipping forward off my ears. I didn't realize. And when I moved my earphones, suddenly I could hear you perfectly clearly.
Okay. Good.
I think that, first of all, we're going to have to get the right computational level story. And my book is a long argument that we don't. The computational level story that we've been using to search for the the physical basis of memory in the brain is the wrong story, and it's causing us to bark up the wrong tree. There are no raccoons up that tree. And as long as we go on with the wrong computational story, we're never going to find what we're looking for, because we're looking for something that doesn't exist. So the book says, well, what should we be looking for? And the book argues that, we're looking for memory, and we're looking for memory in a device that we agree is a computer. And these days, most of us have bought into the computational theory of mind. We believe that our computational level story is going to involve literally computations, that the brain is... There's pretty general agreement about that, not universal, but pretty general. do we know something about memory in computational machines? Well, yes, we do. I mean, go over to the computer science department and ask them, and you'll get an earful. Computer scientists have an understanding of why computers have memory, right? And why they have the kind of memory that they have. And every person who has tried to build a computing machine, going back to Babbage, going back a couple hundred years, has basically ended up doing it in the same way. And they've always created a read-write memory. And all modern computers have a read-write memory. And even when they're talking about quantum computers, they're not talking about doing away with a read-write memory. Far from it. And this isn't a technological accident. It isn't because we got stuck with computers that were built like this, and actually if we went back and started over, we could build them better. I mean, that's just not true. We understand, and the book tries to explain at great length why computers have memory, that memory plays an absolutely fundamental role in computation. And so the kind of memory, this Hebbian synapse, does not appear to be even remotely suited to play that role. And oddly enough, there's agreement about that. That is, many people who believe in the Hebbian synapse, they start right out by saying, well, yeah, brains compute, but they don't compute like computers. Because they have these Hebbian synapses, right, they don't have the kind of memory that a computer has. Well, the book says, look, no machine without the kind of memory that a computer has can be a good computing machine, and the brain is one hell of a good computing machine. So the brain must have the kind of memory that a computer has. This is absolute heresy in neuroscience. I mean, really, this places me completely beyond the pale.
I think that's fair to
say. But I think that I hope, because I really would like to know what the physical base of memory is. I hope over time people will slowly come to realize that, look, it's true. There's got to be a read-write memory. And the Hebbian synapse is not a read-write memory, so we've been backing the wrong horse here, and we should change our ideas about what we're looking for.
So one question I had, throughout reading the book is that you have these just truly beautiful examples, either of work that you yourself have done or the work that other people have done using animals, right? So that's, for instance, I mean, the easiest case probably is the ant case that we've been talking about. And a lot of the arguments are made. I mean, they're supposed to be illustrative in some sense because you think it's the brain of an ant, right? I mean, don't think that this is such a complicated thing. I mean, the brain of ants are doing it,
and the ants diverged from human beings at about the Cambrian, right? So it's likely that the basic mechanism that enables them to do it has been with us now for 500 million years.
Right. Good. So, but then you might also think, well, these are great examples, and I mean, for exactly the reasons that we're saying. But then you might think, well, maybe there's something really specialized about what the ant is doing, or some of the other animal examples that you give. I mean, this is one of the primary things that ants have to do. Maybe they developed some very, very special mechanism that sort of allowed them to have this dead reckoning ability. And then we did talk about, so for instance, if you show the map of what a desert ant, the path that they've gone and whether or not they could work out how to get back. I mean, the sailors will say, no, I really don't think that I could do this. But you might think that, I mean, if it's so easy to implement in a read-write memory in the way we've been talking, why is it that the ants are so good at things that we might actually be bad at in some cases?
Well, I certainly agree that the ants are relying on a special purpose mechanism that evolved for this exact purpose. In fact, that's been another one of my arguments. As you know, the believers in Hebbian synapse also believe that there's only one mechanism that mediates every kind of learning, which I think is terrible nonsense. And dead reckoning is a kind of learning, but it's a very special kind. But the first point to make is that all God's critters have the problem of getting home.
Yeah.
And so we all have this same special purpose mechanism. We all do dead reckoning. I've done experimental work on dead reckoning in human beings. Many human beings think they can't dead reckon, but in fact, everyone we've tested dead reckons rather well. Perhaps not as well as the ant, but the ant's is under huge selective pressure for dead reckoning under very difficult circumstances.
Right.
But everybody dead reckons, so everybody has this mechanism. But that's all of these special purpose mechanisms require memory. I mean, in a computer, my computer is running, probably 150 different applications, each of which is a very special purpose application. But if you took a look away the random access memory in my computer, every one of those 150 applications would be dead. Because they all, this gets back to the fact that memory plays, that read-write memory, which is a well-understood, well-defined notion. It's not some vague notion. Plays an absolutely central role in every kind of computation that any computer carries out. And Adam King's and my argument, Adam King's my co-author in the book, is that there has to be a read-write mechanism in nervous tissue, and it will play a role in every kind of learning. And yes, there will be many very specialized forms of learning, but they will all make use of this same basic memory mechanism, just as all the different parts of the brain send information between one part and another with an action potential, right? An action potential is the universal mechanism in all species, in all parts of their nervous system, that carries information from one place to another. Memory carries information from one time to another time, and that function is just as basic as carrying information from one place to another place. And there's every reason to think that just as there's only one action potential, there's only one basic memory mechanism that carries information from one point in time to a later point in time.
Right. So you might think it's just as fundamental that we're able to sort of store some value as it is that we're able to have information be transferred from this place in the brain to this other place in the brain.
You need to carry information has to move around in the brain, and it has to be carried forward in time. And there's just no way of getting around it. And the story about the Hebbian synapse. So you ask people, so how do you store information in a Hebbian synapse? They won't give you an answer. Well, if they can't even answer that question, then of course they can't answer the question of, well, you can imagine a mechanism where you could store it, but you could never get it back. Right. In fact, in the early days of computers, some of them were like that. You could store it, but you could never get it back. Well, if you can't read it, so if you can't write it, of course, then you have no memory at all. If you can't read it, you have no usable memory, right? So, so you need.
And you might as well not write it.
You might as well not write it if you can't read it. So, it's absolutely fundamental, and the people who are focused on Hebbian synapses just refuse to address the question of how you use the Hebbian synapse to store information. You have to have a code of some kind, right? Every information storage mechanism known to man, there's a code, including, of course, DNA. And so pull out a major neuroscience textbook like Kendall, Schwartz, and I forget who the third author now is, but and look in the index and see whether there's anything that it says about the code by which the nervous system stores memories. I don't think you'll find anything.
Right. Yeah. So, I mean, I think one of the things that's been, I very much like your book, and I really, really enjoyed reading it. But I think one of the things that's interesting is that people have been so resistant to taking up the challenge that you go to such great lengths to lay out, and go to such great lengths to show that they have to take it up if they want. I mean, do you have an idea of why that there's so much resistance to it?
Yeah, I do. It's complicated. There's both intellectual and sociological reasons. Intellectually, it's an enormous break from what everybody has thought since time immemorial. I mean, this story, this synapse story, that was the story when I was a graduate student. That was 50 years ago. And the story was not new then. The people started talking about synaptic change as the basis of memory at the end of the 19th century, right?
Right.
And it just, intellectually, the reason is people have found this so compelling that they just can't bring themselves to believe that the story could be fundamentally, basically utterly wrong, right? Not a little bit wrong that needs a little patching up, needs a little refinement, but just plain wrong. Just like phlogiston, right? Exactly like
phlogiston.
Phlogiston is the famous example of a central concept that seemed completely obvious, right? I mean, heat was this substance came out of things when they burned, right? It was pretty obvious.
Right.
You could almost see it coming out, right? You could feel it. And Priestley and many other chemists back in the 18th century, Lavoisier said, there's a problem here. When things burn, they get heavier. They don't get lighter. That just ran into enormous resistance. The idea that it was actually something in the air that was joining up with this substance, it was such a radical departure from what people had thought. Heat wasn't a substance. How could it not be a substance? So it's just that's one part of the problem. Another part of the problem is that I cannot point to, and believe me, I would like to be able to point to, a plausible mechanism in the nervous system that we currently understand, in which I say, look, this could easily be the read-write memory. It's not that I can't make up stories. People want a story so badly that I've made up several, but the reason I like the genetics story I mentioned earlier about no one getting the right idea is that's why I'm reluctant to push the stories that I make up, because I think the odds are that I'm not making up the right story. But the point was, the people who were trying to make up a story about the gene, they were right that there was a gene and that there was a perfectly comprehensible mechanism that did just what the geneticist said it did, right? And it was a particle, just like, in essence, a particle, just like what Mendel thought 100 years ago, right? And the particle just sat there through whole generations and then re-expressed itself 5 generations down, right? And I mean, the geneticists had it right. The problem was to link what the geneticists were thinking to the biochemistry. And until we saw the structure of DNA, this was a bottomless mystery. And so the second intellectual problem is that at the moment it's a bottomless mystery what the mechanism is. And scientists don't like bottomless mysteries. And then there's also a sociological aspect to it. I mean, the people in neuroscience who don't do the neurobiology of memory, I get invited fairly often to talk at their colloquia. Because they've been listening to this Hebbian synapse story for 40, 50 years now, and a lot of them are muttering, this story isn't going anywhere. And it's full of hand-waving and obscurity and so on, and it's not getting better. But the people who are doing it, who do the neurobiology of learning and memory for a living, right, I mean, they've got their whole career sunk in this idea, right?
Right. Yeah.
You know,
Not good to-
Planck remarked, well, this is the way he's usually quoted. It's not quite what he actually said, but he's been shortened to saying, you know, science progresses one funeral at a time.
Oh, yeah, yeah.
And in your generation, there are a fair number of people who are starting to say, you know, maybe we should rethink this.
Yeah.
But people in my generation, or even the generation behind me, they're not about to rethink it. That's too. That's asking too much.
Too much invested. I mean, so and maybe we can end on this question. I mean, do you think that what do you think the likelihood is? I mean, it's obviously hard to know, but do you think it's likely that say in my lifetime?
I hope. I'm growing very pessimistic that it'll happen in my lifetime. I'm a basic optimist about the long-term progress of science. If I had to guess, but boy, it's a guess, I would think in your lifetime there's a reasonable chance. In my lifetime, with every passing year, the odds get longer. But people keep plugging away at these mysteries. I mean, after all, many of these things have remained mysteries for centuries. It's astonishing what scientists have been able to figure out about all kinds of things. And this is a huge challenge, but I'm convinced sooner or later we'll do it. But it's going to take a while.
Yeah, and it would be, I mean, this discovery would be easily equivalent to DNA. It seems. I mean, the amount of power that it would give us.
This discovery would transform neuroscience. I don't think.
Not just neuroscience. I mean.
Well, maybe not. Philosophy and many other things. But the same is true for Watson and Crick discovery. I mean, I think people who are outside the life sciences don't grasp how profound, how revolutionary, how many things have flowed from the Watson and Crick achievement.
Right.
And it created molecular biology, right, which is sort of biochemistry by a different name, but which has different intellectual foundations from biochemistry, and it absolutely transformed biochemistry. And so, I mean, if a biochemist from the 1940s wandered into a modern symposium, he would think that he would wonder, what happened to my discipline? I mean, I have no clue what these people are talking about. Right? There's a supposedly respectable chemist up there, and he's talking about an error repair mechanism. So on. So it would be just knowing the physical basis of memory would be transformative in neuroscience for just the reasons we argue. It's an absolute foundation of the brain's computational capacity. Yeah.
And so I guess we can hope that in 40 years, the people doing neuroscience today, if they walked into a neuroscience symposium, they too would.
One can hope. I do hope.
Okay, good.
Luckily, I come from a long-lived family, so I hope I may live long enough to see it. But
I hope so too. I hope we're able to work out these problems. It was absolutely wonderful talking to you, and thanks so much for being on and being willing to do an interview and be on the show.
It's a pleasure.
Okay. All right. Take care.